Good morning, my dear brothers. Good morning. Welcome back to our Biomedical Ethics class. Of course, this is our last class. In our previous class, we began with stem cell research. No? We began with stem cell research, and we saw three video clips, that gave some basic idea, basic knowledge about what is stem cell therapy or what is stem cell research. Okay, now we shall go to theory now. What is, I shall make it very short. What is stem cell research and what does the church say? And immediately after that, we have another chapter named cloning. That also we see in briefly what does the medical terms, definition and a few cases. Also the first what you call test tube baby, the sheep dolly. Then the implications of cloning and what does the church say in a short and nutshell. Okay, and perhaps this is our last class. Okay, this is our last class. Good, we shall begin. Now, what is stem cell or what are stem cells? Our body, our human body is made up of many different types of cells. There are many cells and what you call different types of blood, tissues, skin. We have many cells in our different parts of our body. All the organs, they are carrying different cells. Without cells, we cannot think of our life, that which communicates from the nerve system. Even the nerve system, there are cells. Everywhere, cells are filled up in our human body, so that which carries sometimes the message, carry the energy or the blood. There are sufficient work that they are given to perform our human activity and what you call our human life. Good. Now, most cells are specialized to perform particular functions. As I said, each cells or what you call system has got their own functions to execute in our body. Depends on the command that is being given from the from the brain, from the mind. So they carry out all the commands and the functions that are given by the mind. Such as red blood cells that carry oxygen around our bodies in the blood. The red blood cells which are there in our body, they are given the the work of carrying the oxygen around our bodies in the blood, but they are unable to divide. They can't be divided. Okay, they cannot be divided. So stem cells provide new cells for the body as it grows and replace specialized cells that are damaged or lost. So when we meet with an accident, or there is a case of a burned, my skin or my part of my body is being burned. Now that particular part, if I want to replace with the new skin, with the new tissue, with the new cells, now people use a stem cell therapy. But then it is very costly affair. It is not that easy, not affordable, but things are there, but it is costly. Now, they have two unique properties that enable them to do this. First is they can divide over and over again to produce new cells. Okay, stem cells could be produced over and over to new and different forms or different types of cells. Again, as they divide, they can change into other types of cells that make up the body. Okay. Stem cells are special human cells that are able to develop into many different cell types. This can range from muscle cells to brain cells. Could be any cells that starting from our muscle, maybe from our thigh, from leg, or from hand to the brain. Any part of a body, wherever the cells are placed, they are functioning. This can range from muscles to brain cells. In some cases, they can also fix damaged tissues, not only cells. Also, it could be replaced to those areas, those places in our body due to injuries or damage, loss of skin, etc., etc. Even tissues could be replaced with the stem cells. Okay. Researchers believe that stem cell based therapies may one day be used to treat serious illness such as paralysis and Alzheimer disease. So far we have not come to that stage, but then in the future, the scientists believe that even those who are down with the sickness, the serious sickness of what you call paralyzed and what you call Alzheimer, when they could be treated, what you call those cells which are died and gone away, could be replaced with the new stem cells. There is a possibility. It's only the future, what you call vision is there. So far nothing has taken place, but then they are trying, researchers and scientists, they are trying their best to what you call to cure, to heal these incurable diseases like such as Alzheimer and what you call other diseases. In the future, they may be used to replace cells and tissues that they have been damaged or lost due to the so-called diseases that we are talking about. Now, I don't know how far you could see image. These are small stem cells. From there, it could be divided into more stem cells. This is one single stem cell. From here, it has divided into many stem cells. Also, from here, it also could be divided into many other specialized cells. Not the same, could be also divided into some other specialized cells, depends on the what you call the cells that are placed in our organs, in our body. So it has got, each part has got it's what you call special cells. So stem cell could be divided into more cells, also it could be divided into what you call other specialized cells. Again, here another we have picture about the embryonic stem cell. You know what is embryonic, right? They take what you call from man and woman, they take and do it in the IVF. So embryonic stem cell, that is what you call when that could be also divided. The same way we also have adult blood stem cells that is taken from bone marrow. That is taken from bone marrow. What? The adult blood stem cells. Again, they are what you call divided into different types of blood cells. Stem cell therapy is a procedure that uses stem cells to treat a disease or condition. Currently, stem cell therapy treats diseases and cancers of blood. Stem cells are unspecialized or undifferentiated cells that have the unique ability to give rise to many different cells, such as cells in the skin, cells in the liver, cells in the kidney, heart, neuron, in other organs of what you call a human. They also possess the property of self-duplicating. It's a copying from one to another, self-duplicating for indefinite periods of time, and are thus present as adult stem cells in most of the organs of the body, including blood and bone marrow. Now, let us see what are the what you call different types of stem cells. Again, we have three types of stem cells. They are embryonic stem cells. I have put a short form ESC, embryonic stem cells. Second, we have adult stem cells, ASC, adult stem cells. And third, induced pluripotent stem cells. What you call the abbreviation comes IPS, not Indian Police Service, but induced pluripotent stem cells. Let us see now each one. What do they say about? First, embryonic stem cells. Embryonic stem cells supply new cells for an embryo as it grows and develops into a baby. When there is a defect, when there is a the embryonic cells are down with some sickness, or they are not that good enough, healthy enough, they could be replaced with other what you call embryonic stem cells. As they grow, maybe what you call the womb, or as they grow in vitro petri dish, they could be done a research and they could be easily replaced. These stem cells are said to be pluripotent. Now, what is pluripotent? Pluripotent is that which means they can change into any cell in the body. That is what you call the previous picture we have seen. Induced pluripotent stem cells. Pluripotent would mean that could change into any cell in the body. They can divide, they can multiply even. They could be divided and multiplied. into any cell in the human body. So this is a embryonic stem cell. Okay. Second, adult stem cells. Adult stem cells supply new cells as an organism grows and to replace cells that get damaged. I said, when part of your body organ is being damaged, being lost due to burning or due to accident, due to some sickness, due to some cancer, so they could be what you call easily replaced, those damaged cells in the body. Adult stem cells are said to be multipotent. Okay, they could be easily multiplied in many numbers. Okay, which means they can only change into some cells in the body, not any cell. Okay, they could be multiplied, they could be divided, not what you call any cell, that particular category. Okay, that particular category of that particular organ or what you call a particular place. Now, for example, blood, that is what you call hematopoietic, that is pertaining to the formation of blood or blood cells. So blood stem cells can only replace the various types of cells in the blood. The blood stem cells cannot be replaced with other cells that are in other parts of the organ. So blood cells could be used only to the particular what you call blood cells. Cannot be replaced with other cells which are in other parts of our human body. Skin, another example. So skin stem cells provide the different types of cells that make up our skin and hair. Okay, skin and hair, what you call those cells are different from what you call other cells. For example, the blood cells. Blood cells cannot be replaced with the cells that are connected with our skin or that which gives nutrition, food to what you call our hair. It has got special cells that are on our skull. It has got special cells, and of course they are also what you call partly connected with the blood, but they are not the same. It has got different functions and they are not one and the same that are in our what you call other blood cells. Okay, clear? So first is embryonic stem cell, that you understand, the embryos that are taken, done research, okay, in IVF, okay, embryos, the fertilized egg, that is first is embryo. Second is adult, that is taken from the what you call mainly the bone marrow. In the previous picture we have seen, you remember? Bone marrow, adult blood stem cells. Okay. Now third, we have induced pluripotent stem cells. First is embryo stem cells, second is adult stem cells, and third is induced pluripotent. Pluripotent, as I said, they are capable of giving rise to several different cells. They could be easily multiplied, they could be divided into more. Okay, capable of giving rise to several different cell types, that is a pluripotent. Now, induced pluripotent stem cells are IPS. I have put I said it's not an Indian Police Service, but induced pluripotent cells are stem cells that scientists make in the laboratory. Okay, induced means that they are made in the lab. Induced means that they are purposefully made in the lab by taking normal adult cells from the what you call so-called the bone marrow. Adult cells are taken from the bone marrow and they are made up in the purely in the lab. Okay, the research center. Like skin or blood cells, and reprogramming them to become stem cells. The cells, what they take, what you call, from the adult stem cells, they work on it, in the lab, and they can be, reused or reprogrammed as a stem cells. Because it's a big process. It is not a one day work or half an hour work. It's a gradual process that they have to work on it. A lot of research has been done. Just like embryonic stem cells, they are pluripotent. So they can develop into any cell type. This is what you call some basic information about what you call stem cells. Of course, there are much things to be, go deeper, but we don't go so deeper. It's only what you call basic course, and we just know the basic knowledge of what is stem cells. Now we understand, So stem cell could be what you call from the embryo, no, that is better fertilized egg, could be what you call the adult, no, adult stem cells that is taken from the man's what you call bone marrow. And third, we have induced, pluripotent that they are taken and they are working in the lab. They could be multiplied, they could be reprogrammed or reworked so that they become another stem cells. Okay. Now let us see what the church says with regard to stem cell research or stem cell therapy. Church on what you call stem cell research. Catholic teaching respects the dignity and the equal moral worth of every human person. This view has consistently rejected research that destroys the life of some in order to enhance the life of others. No, you cannot kill somebody so that you are giving life to the other man. No, that cannot be. No. You want to save one man, so you have to kill another man. No, that church doesn't tolerate, church doesn't allow. Okay, this idea, this concept. The church affirms the position of modern embryology that the human embryo is a living. No, embryo is a living, complete, whole and integrated, self-directing member of human species who will, if given the proper environment, move itself along a trajectory of development to the next mature stage. Sure, embryos got the life, to become a human person. Each embryo has got the life, has got the what you call a dignity to become a human person. Only size is small, a small cell. When it is given what you call its environment, it stands to grow. Sure, it will grow. In 9 months, it's going to be a baby, 9-month-old baby. In 2 years, you are going to be a what you call a small little, in 15-20 years, an adult. So it has got equal respect to any other human being. The Catholic Church opposes research involving human embryonic stem cells. Opposes only human embryonic stem cells, which require the destruction of the human embryo during their isolation. Okay. In contrast, the church has championed, or encouraged, gives the permission to adult stem cell research. The church doesn't say anything against adult stem cell research, whereas against the human embryonic stem cell, it is against. It will not encourage against, because human embryo has got the life to become a human person. Okay. In contrast, the church championed that adult stem cell research which does not involve human embryonic stem cells. Additionally, the recent breakthrough to program somatic cells into embryonic like induced pluripotent stem cells has further diminished the need to utilize the human embryonic stem cells for research and medical therapies. When they found what you call this IPS, induced pluripotent stem cells, they could be worked on, they could be reprogrammed to use as stem cells. The need to use embryonic stem cells has been reduced. So people stopped taking or using embryonic stem cells, and they began to use only what you call induced pluripotent stem cells, because they could be worked on and they could be transformed into any other stem cells. They could be matched in what you call other parts of what you call a body. So the usage has come less. Okay, which one? The embryonic stem cell. As the US Conference of Catholic Bishops declared, clearly the Church favors ethically acceptable stem cell research. It opposes destroying some human lives now, on the pretext that this may possibly help other lives in the future. We must respect life at all times, especially when our goal is to save lives. This is what you call a Catholic Conference of Bishops of what you call US Council, and they came forward with regard to what you call a church view on what you call in favor of stem cell research, but not embryonic stem cells. Okay, because it kills the human life which is in the form of small cell. At the same time, in the instruction Dignitas Personae on certain bioethical questions, that is Donum Vitae, of the Congregation of the Doctrine of Faith, declared, the Church therefore views scientific research with hope and desire. That many Christians will dedicate themselves to the progress of biomedicine and will bear witness to their faith in this field. Research initiatives involving the use of adult stem cells, since they do not present ethical problems, should be encouraged and supported. Okay? The research involving both adult stem cells and induced pluripotent stem cells that do not require the use and the destruction of human beings at any stage of development, for the sake of the common good. In addition, there are many researchers who believe that adult stem cells and what you call IPS, induced pluripotent stem cells, may provide a greater benefit for the development of therapies for debilitating human diseases than the human embryonic stem cells. Okay, when the scientists found that instead of taking the embryonic stem cells, they take from adult or they take induced pluripotent, the usage of embryonic stem cells has come down, and with that, with the stem cells of what you call adult stem cells and the induced pluripotent stem cells, they could be reworked and reprogrammed and could be used in other way, so that the human embryos are being saved. They are not destroyed. They are not destroyed. The Church objection to some forms of stem cell research is expressed in Donum Vitae number 4. Medical research must refrain from operations on live embryos. There should be they should be stopped. Those operations, those researches on human live embryos, unless there is a moral certainty of not causing harm to the life and the integrity of the unborn child, which is in the form of small embryo, and the mother, and on condition that the parents have give us their free consent and informed consent to that procedure. It follows that all research, even when limited to the simple observation of the embryo, would become illicit were it to involve risk to the embryo's physical integrity or even the life by reason of the methods used or the effects induced. As regards experimentation, and presupposing the general distinction between experimentation for purposes which are not directly therapeutic and experimentation which is clearly therapeutic for the subject himself. In the case, the point, one must also distinguish the difference between the experimentation carried out on embryos which are still alive and experimentation carried out on embryos which are dead. Now, we all know so much of cells and embryos, they are taken and use only what you need, and you keep on researching them. You don't use it for the benefit, or when they are really, or they are damaged, or they have to be given care, or they have to be worked, and they have to be given proper medicine in order to have life. That is one aspect. But when they simply work, do experimentation and destroy them without any proper reason, or without even even the consent of the couples, mother or even the father, we have to inform them that they are going to use for the experimentation's sake, because it's not allowed. Ethically, it is not allowed, church view, but then they take, sometimes they do it when without informing the couple that, see, they can take and they can manipulate, they can misuse, they can insert in some other couple, and they can claim, this sperm belongs to this couple, and there could be misunderstanding, lot of confusion. So usually we have to avoid using any experimentation so that they are not misused. They are not misused, or they are not simply destroyed at the lab. That is what the church would want to say. If the embryos are living, whether viable or not, they must be respected. Whether we use them or not, they have to be respected, just like any other human person, because they carry They carry equal status of the human person. Only the structure is different. They are small. We the human beings are big in our physical structure of our nature. Experimentation on embryos, which is not directly therapeutic, is illicit. No objective, even though noble in itself, such as a foreseeable advantage to science, to other human beings, or even to the society, can in any way justify experimentation on living human embryos or fetuses, whether viable or not, either inside or even outside the mother's womb. Okay, this is what you call Congregation of the Doctrine of Faith will say about what you call importance on the dignity and the respect of human embryos, which are simply destroyed or which are simply misused in the lab during what you call a embryonic stem cell research. So the church is again completely against embryonic stem cell research, but not against other what you call adult stem cell research or what you call induced pluripotent research. You get me what I want to say? Okay, church is only against the research which destroys, the human what you call embryos. Okay, this is what in a nutshell I have prepared about what you call stem cell therapy, stem cell research. Of course, we have a lot of information from those 3 clips we have seen. Okay, if you want, we can again rewatch those 3 video clips so that you have more idea. Okay, how what you call biologically they are connected, on how the scientists and the researchers they would use it in the modern world. Okay, is it clear? Oh, good. Now we have completed this chapter, so called stem cell research or stem cells. Now we shall move to cloning. Okay, we shall move to cloning. So as we begin, we shall see 2 video clips about the origin of cloning. Okay.
So, what comes in your mind when you hear the term cloning? If you're like me, then you might be thinking of the Clone Warriors from the Star Wars movie, right? Now, let me tell you that it's not only Clone Warriors, but cloning could be done for anything, such as DNA. There could be clones of DNA, there could be clones of cell, there could be even clones of this cat. In fact, the identical twins are clone of each other. That simply means clone are exactly similar things, right? Now the question is it possible to make a clone of an animal? Sounds pretty sci-fi, right? And it turns out it is possible, and it was possible because in 1996 there was an experiment by Prof. Keith Campbell and Ian Wilmot, and the experiment took place in the Roslin Institute, Scotland, and they cloned Dolly. So introducing Dolly the sheep. So why Dolly is so special? It turns out Dolly is actually a clone of a Finn Dorset E. Now Dolly the sheep was pretty popular. Even still now she is pretty popular because she was fertile, she can give rise to offsprings, and she was exactly same to her mother in terms of genetic makeup. So let us try to understand that how Dolly was created. So here is the mother of Dolly, who is a Finn Dorset E. Now from Dolly's mother, scientists took the udder cells, which is the skin cells from the mammary gland. Then from Scottish blackface E, they took the egg cells. So the Scottish blackface E work like an egg donor. Now they cultured the somatic cells from the Finn Dorset, and this somatic cell culture would be used later on. But from the Scottish blackface E egg, they took out the nucleus with the help of a pipette. And these eggs now become enucleated. So note that there would be no genetic material of Scottish blackface E in these enucleated egg. Now they use electric shock to fuse these two cell types, the somatic cell and these egg cells coming from the black-faced eel. Now there would be a hybrid cell type. Notice that in this hybrid cell type, the nucleus or the genetic material would be totally similar to the somatic cell donor, that means the fin dorsate, whereas the cytoplasmic material would be a mix-up between the two. Now these cells would ultimately form an embryo and they would develop gradually. Now when the embryo reach a blastocyst stage, it is ready to be implanted to a surrogate mother. In this case, it is implanted to a Scottish black-faced eel. And when the progeny was born, surprisingly enough, the progeny was exactly similarly looking like the fin dorsate eel. And they call it the Dolly. Dolly was the name given to these newborn lamb. Now, Dolly was the clone of fin dorsate eel. Let me remind you, this fin dorsate eel work like a somatic cell donor. So they are pretty much similar in terms of their genetic makeup, right? Now Dolly was fully viable. When Dolly grew up like her mother, she gave rise to Bonnie. Bonnie was the progeny of Dolly, and again Bonnie was exactly similar to Dolly. And yep, so Bonnie was born out of Dolly. So Dolly was fully viable, but eventually Dolly died. Now let me tell you that cloning can bring industrial revolution. So let us talk about the plus and negative point of cloning. So let's think from the industrial point of view. So let us understand that we have a cow farm, and these cows are used for milk or meat purposes. So let's say there is a cow which gives a very good quality of milk and very healthy, so it's a good source of meat as well. So this cow is a star of the farm. We wish that if we have multiple of these cows, then it would be really beneficial for the farm industry, right? Or the meat industry. So in theory, we can generate the clones of these star cow, and that would give us optimal quality meat and milk. Imagine how beneficial it would be for the meat industry or the dairy industry, right? But there are ethical concerns. In fact, scientists tried cloning in cows and also in pigs, keeping these things in mind. Obviously, the cloning was successful, the progeny was born, but due to some unknown reason, these progeny were more susceptible to infections and they are dying. So it was expected that they would lower the cost of farming and they would really give rise to optimum quality of meat and milk at low cost, but their maintenance was way higher. So still it's a problem. Now, while scientists were cloning cats, some dramatic came out of that. So this is a calico cat which was cloned, and her clone was genetically exactly similar, but look at her coat color. So identical DNA, but not non-identical cats. How that is possible? That means even if two organisms have exactly similar genetic makeup, they might have differences, and these differences might occur at epigenetic level, beyond genes, right? So clones might not be exactly similar. Now let us imagine the problem of cloning and let us try to understand. So here is a pig population. Different colors represent different genetic makeup of these pig, though they are same species, but they could be having different gene expression profiles, and that makes them a bit different from each other. Now if you start doing cloning over a time period, there would be exact similar genetic makeup in a population. For a population, it's very bad because it causes inbreed depression or it increases the risk of genetic disease. That really questions the plus point of cloning, because it ultimately do not create genetic diversity, or it reduces the genetic diversity, which is very bad from an evolutionary point of view. So, but there could be benefits. Think about the endangered animals. We know Royal Bengal tigers are endangered. Their numbers are very less. One-horned rhinoceros, polar bears, elephant, they're all endangered. Only few thousands of them. If we don't take care of them, if we don't conserve them, then they would be totally extinct from this earth, right? And they would be only left in the pages of books. So using cloning method, we can increase their population. So let us imagine this is a Royal Bengal tiger. We can make a clone of this Royal Bengal tiger, and that is how we can really increase their population, which is now decreasing. You know, in a small population, there are problems like genetic drift and allelic fixation, etc. So we can rescue them from that small population. But again, when the population would grow, it would be a homogeneous population of a tiger. That would create a huge problem. That would, in turn, increase the risk of genetic disease. So, well, this problem is pretty complicated. Even scientists tried this cloning method to make human organs inside a pig, but that eventually did not lead to success. Now, whether cloning is good or whether cloning is bad, it is an alarming research ethical topic, and I can't really comment more on that part. But anyway, in this video, we looked at what is cloning, how Dolly was made, possible benefits of cloning, and possible downsides of cloning. That pretty much summarized this topic. I hope you enjoyed this video.
Dolly the sheep was a sensation when she first appeared before the world. A scientific breakthrough, fascinating but also menacing. Dolly was cloned by taking a body cell from sheep A, fusing that cell's DNA with an egg cell with its nucleus removed from sheep B. This fused cell developed into an embryo, which was placed in the uterus of a surrogate mother, sheep C. The resulting lamb, in this case Dolly, is a clone of sheep A. These were Dolly's so-called siblings, identical sisters, all cloned from the same sheep. Professor Kevin Sinclair worked with them and knew Dolly.
It was a step change in our understanding of early development, which ultimately led several years later, in 2006, to the discovery that you can actually induce somatic cells, these are sort of cells that belong to the body, to become embryonic-like cells that has the potential to divide into other cell types.
This was Dolly's greatest legacy, advances in what we call stem cell research. The ultimate, but still elusive goal, is to use cloning technology to create healthy tissues which can be transplanted to heal damaged cells and organs. But back in 1997, Dolly seemed to raise more frightening issues. If we can clone a sheep, many asked, why not a human being? Since then, scientists have cloned more than 20 species, including horses and household pets. But scientists have also recoiled from using these technologies on humans.
I think where we draw the line is if we're going to use genetic manipulations, we have to be extremely careful, and we have to look for severe disease effects to control for. Because with a lot of these things, it's not just the person you're doing a genetic manipulation on or potentially cloning, but it would affect all the generations ahead.
So Dolly's brave new world has not quite turned out as predicted. A sheep created like none before her, but she could not escape her own mortality. She died in 2003, now stuffed in a Scottish museum. Barnaby Phillips, Al Jazeera, London.